thanks for the opportunity to be here. And I know there's a lot of interest in New Zealand. We just came from Australia, and they're also starting to get concerned. But here in New Zealand, you guys have been much more on top of some of these invasive species. And I know we've been working with Gonzalo now for a little while on some of the behavior and biology of one of the key natural enemies of brown marmorated stink bug, and hope to be able to continue this collaboration. So, as Gonzalo mentioned, I'm going to be talking mostly about the biocontrol agent that we work with for brown marmorated stink bug, but I was also asked to include some comments to give an update on the situation with these other invasive species, spotted winged Drosophila and spotted lanternfly in the U.S. So, and I'll get back to that toward the end of the presentation. Also, as Gonzalo mentioned, our lab in Newark, the Beneficial Insects Introduction Research Lab, we focus on classical biocontrol of invasive insect pests that get into North America. So my projects all have to do with agricultural and landscape pests, but we also have scientists that work on invasive forest pests like emerald ash borer and Asian longhorn beetle. For those of you who may not have seen pictures of brown marmorated stink bug in a situation where it can get bad, this is a good example. These are from the mid-Atlantic states and the central coastal east part of the U.S. In the fall, brown marmorated stink bug has this aggregation behavior where they come together in response to aggregation pheromone, and they start looking for overwintering sites in protected habitats. Which humans conveniently provide for them. You know, they like to come into houses and offices and sheds and garages and so on to get through cold periods of time. And the picture in the center shows an aggregation on the wall of a bank. It's just tremendous numbers will show up in the same area, and you can see all these spots on the house. You can see all these spots in the house are all stink bugs. They're all attracted to the wall during the sunny part of the day, and then eventually they'll crawl. They'll look for cracks and so on, and they'll go into the house and into the walls and into the attic, where they'll spend the winter. BMSB has been in the U.S. now for several decades, and they were first spotted by the members of the general public who recognized, I haven't seen this bug around before, and they would take specimens to their local extension agent and they ask, what is this? It looks like it's new to us. But it was initially misidentified as a native species, so regulatory agencies and entomologists didn't get too concerned about it, initially speaking, and it took a couple of years for somebody to persist and get it to a taxonomist who eventually did correctly identify it as an Asian invasive species. And that was in around 2000 or so, late 1900s. These were introduced into eastern Pennsylvania. Pennsylvania is a pretty big state. It's a important fruit growing area, and it's a port, and it's thought they probably came into North America in shipments of goods that came up the Delaware River in containers. But it took a few years for populations of stink bugs to build up to the point where people started to notice it's becoming an agricultural pest. And that was in around 2008 or so. So it was in North America for a good 10 years before populations were large enough to become evident as a problem. And then once the first problems started to become evident in agriculture around 2008, very, very rapidly the populations began to build at that point. And we had these years, 2010, 2011, 2012, where growers were just really hammered with stink bug populations. And that's really when the research in North America started on management of brown marmorated stink bug. So we've been working on brown marmorated stink bug now for a few years already, and BMSB is still regarded as an important pest. This is the area where it was first introduced in North America, and it spread, and the initial problems were in this part of the country, but gradually it's spread across the entire country. There are still a few states where it's not reported in any numbers, but most states report it. And the states in red or orange It's regarded as a serious agricultural pest, and the states shown in orange, it's regarded as an agricultural pest, that not at the same levels as in the red states, but it continues to increase in some of the areas where it hasn't been located as long. For example, in the southeastern states down here and in California, populations and impact is still growing. So we still don't know the full extent of BMSB as a problem in North America. Now, most of you probably know that BMSB has a very, very wide host range, which is what makes it a landscape level pest. Populations exist in quite a large variety of native plants, in ornamental plants, in vegetable crops, as well as tree fruit crops. So in any landscape there usually are a variety of hosts to maintain the populations. And they'll build up in unmanaged habitat, and then they'll move into crops. And then they'll move out of crops back into the unmanaged habitat where it's difficult to control their populations. One of the first things we do with an invasive pest is to start to keep an eye on are there any native natural enemies that will attack the invasive species. And we have good information now from a decade or so of surveys in North America. There are a number of native generalist predators and parasites that will attack brown marmorated stink bug, but not at levels that exert significant control to help manage the populations. So the overall level of attack by natural enemies only accounts for about 20% of the mortality of stink bugs on average, and that's not enough to keep the populations from becoming pests. Initially, people were hopeful that some of our native natural enemies would adapt to BMSB and we would see increasing levels of attack, but that hasn't really happened. We've been monitoring in Delaware now for over 10 years, and we see basically the same level of attack by stink bug parasites and predators and parasites, and most of the stink bug eggs that are laid hatch successfully into stink bugs. We're not seeing the native natural enemies adapting. So this led us to start looking in the native range, which is northern, north-central China and South Korea and Japan. And we've been doing surveys in that area now for a number of years, and we have a pretty good idea of the natural enemy complex and what helps to keep the stink bug from becoming as large a problem in Asia as it has become in the invaded areas, North America and now also Europe. And we think that the main mortality factors from natural enemies are egg parasites. And there are a number of species of parasitic Hymenoptera that attack BMSB in Asia, but the most important one is Sulcus japonicus. Throughout the entire range of where BMSB occurs in China and Japan and Korea, Sulcus japonicus is the dominant parasitoid. We also know that Sulcus japonicus is not exclusive to brown marmorated stink bug in Asia. It does attack some other species of Chinese stink bugs. These are some examples. These all happen to be pests. We know very little about the full host range. Our surveys in China are designed, are bringing us more information about the actual host range. And I think of Sulcus japonicus as a specialist in Pentatomidae, but there are a lot of species in Pentatomidae, and within the family Pentatomidae, or the superfamily Pentatomoidea, Sulcus japonicus is what I would call oligophagous. You know, it attacks some species. It does not attack all pentatomids equally. Some of them it doesn't attack at all. Some of them it attacks at low levels. And there are a few species where it's also the dominant natural enemy, such as this species here, Ethicena fullo, which is also a very important fruit pest in Asia and subject for concern for potential introductions to other areas. So Japonicus is also an important parasitoid of that species. Now this kind of work couldn't really proceed without the close involvement of taxonomists. the group Sulcus and the other groups of parasitic Hymenoptera that attack stink bugs in Asia are the status of taxonomy at the start of our project was not sufficient to let us correctly identify all these species. So we recognized that, and we engaged Elijah to begin working on the taxonomy of this group. This is extremely important in a biocontrol program of this nature. And it's allowed us, for example, to the work that's been done by Elijah and his colleagues to help us correctly identify not only the species of egg parasites that we're finding in our surveys from Asia, but also throughout North America. And it allowed us to correctly identify Sulcus japonicus as Japonicus because it was originally described as a new species and given an incorrect name as haliomorphy, which is you may see in the literature. It also has enabled the detection of new advent species, and Elijah will say a little bit about this when he gives his talk. We have also molecular phylogeny of Sulcus now from Asia, and this gives us additional information that some of these species of Sulcus have a good degree of specificity. The ones that are circled in yellow, Metsucuriae, Cultratus, and Japonicus, are all species that we know will attack and develop in brown marmorated stink bug. The other species groups, not so much or not at all. So this helps support our contention that Japonicus is, has some degree of host specificity even within Pentatomida, and this is important for regulatory purposes when we want to obtain permission to release. So these are some of the characteristics of Sulcus japonicus that make it a good parasite. Well, the slide says make it a good inventive parasite, but they also make it a good natural enemy of brown marmorated stink bug. When females find an egg mass, typically they'll attack every egg in the egg mass. Not all species of egg parasites do that. There are some of the chalcidoids that attack egg masses will attack just a few eggs, but when Japonicus attacks it, they'll usually kill every single egg in the egg mass. And BMSB typically has 27 or 28 eggs in every egg mass. So when they're found by Sulcus japonicus, they're all killed. In Asia, the parasitism can easily reach 60, 75%, and as high as 90% or more by the end of the season. And this is much, much higher than the levels of parasitism that we typically see with Sulcus on native species in North America. And this is one reason why we think Japonicus is going to have a good impact on BMSB once it finally gets established and reaches the full extent of its range and dispersal. Sulcus has an interesting biology in that they sibmate. Within each egg mass, the parent female will lay some female producing eggs and some male producing eggs. And the males develop a little bit faster. And they emerge first and they wait for the females to emerge and mate with them right away, so that when the females come out and disperse from their parent egg mass, they're already mated and ready to lay fertile eggs in whatever stink bug egg masses they find in the environment. So it's an ideal life history to have for purposes of dispersal and spreading. And within each egg mass, typically a majority of the eggs produced are females. The parent females will guard their egg masses for a day or so to help fight off competitors. They have a much shorter generation time than the stink bugs themselves, so in each season there could easily be a number of, as many as 8 to 10 generations of parasites produced in a given crop season. The females for small parasitic Hymenoptera are very robust little parasites, and they're easily capable of living for several months if they're provided, if they have access to sources of moisture and carbohydrate. So unlike some parasitic Hymenoptera, which only typically live a couple days, these females will live a long time. they survive, we believe, in nature over the winter in tree bark, in crevices and underneath loose plaques of bark and so on. And this helps them get through periods of extreme cold weather. But lab experiments with Sulcus have shown that they're capable of surviving periods of cold weather as cold or colder than BMSB. So wherever BMSB occurs, Sulcus japonicus is also capable of occurring and surviving. These charts just show the lifespan. The maximum adult lifespan in lab tests was nearly half a year, 155 days. But the typical lifespan is a little over 2 months for a female, a little bit less for male. The females typically live about a week, about 2 weeks longer than males. So they can mate several times, conceivably. And we think that their fecundity is governed partly by their mating. If they only mate once and they continually find egg masses and lay eggs in, the sex ratio will gradually become more and more male-based. But if they mate again, that moves the reproduction back up into the female side. And again, under summer conditions, summer field conditions, the average development time of the immatures from egg deposition to emergence of an adult is about 2 weeks. So they can turn over a generation pretty quickly. In Asia, one of the trees that they've been found under in pretty regularly are these Zelkova trees. But we know that they also can survive under other sorts of trees. Zelkova are very common in Asia. It's a tree that has a large cultural significance, so they're very widely planted. And you can see the bark is very flaky, so there are a lot of places for the parasites to crawl under. So this is some Climax work done by John and Gonzalo, I believe, showing where Sulcus japonicus could likely survive in other parts of the world. And you can see from the charts in Australia and New Zealand that there are areas where Climax indicates the climate is certainly quite favorable. The diagram down in the right-hand corner shows North America, and the circles show where Sulcus japonicus has been shown to have established adventively. So this area, this part of the U.S. is where BMSB first became established and where we first found the adventive populations of Japonicus. Since then, there have been populations found in other states and also in these parts of the West. So even though the diagrams indicate that that might not have been optimal climate for Japonicus, they're certainly perfectly capable of surviving in that habitat. And again, I think in North America and probably in Europe and wherever else Japonicus may establish, they're very likely to be able to survive and do well wherever BMSB occurs. Now, because Japonicus is an exotic insect, just as in New Zealand, in order to receive permission to establish an exotic species in North America, we have to go through an extensive series of host range testing. And before Japonicus was found as an inventive species, we had begun these evaluations with a population of Japonicus from Beijing, which we call the Beijing strain. We have Japonicus populations from many locations in Asia, but we started our evaluations with that particular population because it had been described in literature and it had a name assigned to it, which is an advantage when we're preparing a petition. And these host range evaluations were designed to show the physiological host range of Japonicus when exposed to a variety of non-target species. And the diagram, or the photo on the top left corner, shows the exposure arena we use. This is a plastic vial about this long and about an inch wide, and each container had one egg mass of either a brown marmorated stink bug or one of our non-target species. And we always expose the same female to both the non-target species first and then to the BMSB to show, to be certain that that particular female was competent. Sometimes we would have a female that couldn't attack anything. It wasn't healthy or whatever, and those were replicates that we were able to discard. But we wouldn't have known that if we didn't have the positive control. So in Delaware, we tested about 25 species of native North American pentatomids or resident introduced species like this, well, not Nazara. We didn't test Nazara because we don't have that in Delaware, but other introduced species, most of them natives. And this diagram shows the results of our no-choice tests. The purple bars shows the performance of the female on brown marmorated stink bug compared with the bars in orange, which shows their performance in the non-target species. And this performance is measured as emergence of a healthy F1 generation offspring. And you can see that in very few of the non-target species supported full development of the wasp. Only a handful, 4 or 5 of them produced a significant number of adult wasps. So this indicates at a physiological level that something is going on. There's a fair degree of specificity. However, if we compare this with the number of non-targets that produce healthy unemerged or healthy stink bug nymphs, we can see that there's a big difference. In some of these, you can see that there were no wasps produced. Whoops. You can see that there were no wasps produced in the non-target species, and the stink bugs emerged in good numbers. So there was no impact from Sulcus Japonicus. But in many of these other examples, you can see that there were no non-target, that there were no Sulcus emerged, but there were also no non-target healthy nymphs emerged. And this indicated that the wasps were stinging the non-targets and killing the nymph, killing the non-target host at some stage in its development. So there was impact, but the impact just didn't lead to the successful production of a parasite. And we had cooperators in different parts of the US that tested a wider range of non-target species in their areas. So we had cooperators in Florida, in Michigan, in the central part of the US, and on the west coast. All told, we looked at about 65 species of non-target pentatomidae in North America, and the results were very much similar everywhere. Very few of the species produced an F1 generation of wasps, but there was a fair degree of attack, nevertheless, that resulted in mortality of the non-targets. And similar results were found when Sulcus japonicus was tested on some of the native stink bugs in China that it typically is exposed to. So here in the black bars show the production of Sulcus japonicus in 8 species of Chinese pentatomidae, and you can see that there's a range in successful development. We do know also that there are effects on non-target effects on the fitness of a parasite that emerges from a host that may not be a most favorable host. For example, wasps that emerge from small stink bug eggs are very, very small, and they have fewer eggs, and they tend to be less active. They don't seem to be as good at recognizing hosts when they find them again. You can take a small wasp, like this one that came from from a smaller egg, and expose it to brown marmorated stink bug in the next generation, and it will successfully reproduce and produce large wasps again. But the behavior, the performance of that small female will typically result in less eggs and less success in finding another host egg mass. So there are fitness costs of developing in a non-target host. And most of you are aware that similar host range testing has been done by Gonzalez Lab here in New Zealand with the New Zealand species, and with similar results, a wide range in the ability of Sulcus japonicus to develop in some of the stink bugs that occur in New Zealand, with some of the species supporting pretty good development and others to a much lower degree. And as I mentioned, while we were doing the host range testing in North America, we had the unexpected recovery of a Sulcus japonicus. These were discovered in a survey for native natural enemies in 2014. And without the involvement of Elijah as a taxonomist in this program, it probably wouldn't have been recognized anywhere near that early that we had the advent of presence of Japonicus. You know, it would have just been considered, oh, here's another native parasite, nobody would have looked at it. But this was found in Maryland at one of the labs, actually one of the USDA labs, where they've been working on the impact of native natural enemies on brown marmorated stink bug. And our first thought was, oh boy, is this a release, is this an escape from our quarantine population? And we were able to determine very quickly, fortunately, that it was not. I'll say more about that in a moment or two. But that was a big relief that we were able to determine right away that it was a self-introduced example. And every year since then, we've continued to find Sulcus japonicus moving to other parts of North America. So in 2015, it was found on the West Coast. In 2016, more states on the East Coast and the West Coast. 2017, 2018, 2019, every year has been a new state. And in addition, it's been found across the border to the north, initially in British Columbia, but then also in Ontario. And then about the same time, it was found in Canada. It was also found in Europe. So Sulcus japonicus now also occurs in Switzerland and in northern Italy. And we know that this population is another unique introduction from Asia, probably because it's not related to the introductions in North America. And not only is Sulcus japonicus inventive in Europe, but a second Sulcus mitsukurii, was also found just about the same time in Italy. So we have two inventive Sulcus. This just shows the part of Italy where they were found. Northern Italy is a very important fruit production region. And the question arises, how is Sulcus japonicus moving around, or Sulcus species? How are they getting around so readily? There are a couple of potential routes of entry. A lot of plant material is moved around. It could conceivably have come in within parasitized egg masses of stink bugs on imported plant hosts. As I mentioned, that the adult wasps are very robust wasps, and they can live quite a long time, and it's quite possible for them to hitchhike as an adult, land on a person in an airport, get off on the other side of the world a few hours later, or come out of a cargo container. We don't know exactly how any of these populations of Sulcus have arrived in North America because the populations don't match the genetics of the populations that we've looked at in Asia. But these are certainly some of the potential routes. Another potential route is a deliberate introduction, and we don't have any evidence one way or the other to suggest that that could be a possible route. Certainly possible. They may not even have come in on brown marmorated stink bug because since we know they develop in other stink bug species, they could have come in within parasitized eggs on other hosts. And of course, this expands the potential range of routes of entry, as a wider range of plant material could be considered as an introduction route. And again, the characteristics that lead it to be a successful inventive parasite are the same characteristics that we regard it as being an effective parasite, a brown marmorated stink bug. Short generation time, long life as an adult, and so on. In North America now, a lot of our effort has shifted to monitoring for the presence of Sulcus japonicus. Of course, in states where it doesn't yet occur, growers and entomologists and managers are very eager to have it show up. So there are a lot of monitoring schemes. A lot of researchers are looking for it every year. And typically we use two methods. Some of the most important surveys are done with sentinel egg masses. The sentinel egg masses are a good method for monitoring because they give us information on what species of wasp are attacking successfully brown marmorated stink bug. But they have a disadvantage in that it takes a lot of effort in order to maintain a colony of stink bugs large enough to generate a large number of sentinel egg masses. And then the sentinel egg masses are only susceptible to parasitism for a couple of days. You know, they have to be put out when the eggs are fresh. Stink bug eggs under field conditions in the summer in North America will develop to hatching of stink bugs in about 5 days. And once they develop too far towards a young nymph in the egg, the parasites will no longer attack them. So an egg mass is only susceptible to attack by Sulcus for the first couple of days. So that means that the eggs have to be gathered immediately after they're laid, put out into the field, and then brought back fairly quickly. So it's a huge effort to use sentinel egg masses. Fortunately, Sulcus also respond pretty strongly to yellow sticky cards. And a lot of the finds of Sulcus japonicus have been on yellow sticky cards. And the advantage there is they're easier to set out and bring back in. And people like Elijah can identify Sulcus japonicus while it's still on a sticky card, which is a very handy trait. The disadvantage of sticky cards is that there's a lot of bycatch sometimes. But, you can't have it both ways, it seems. So we were fortunate with Sulcus japonicus to be working with a molecular biologist at our lab in France, Marie-Claude Bonne, and she developed a range of primers, of haplotype primers, that could be used to distinguish among different populations, different geographic populations of Sulcus japonicus. So we know that in Asia there seem to be 6 distinct lineages of Japonicus, and all of the US populations and the population found in Switzerland and Italy belong to this, the largest lineage, lineage 6. And this just shows the relationship of different haplotypes of the Swiss and Italian population. Switzerland is here, and it includes haplotypes that also occur in Japan, which is red. Italy also is here. So the Swiss population seems to be most closely associated with Japanese populations. When these are plotted on a principal component chart, we can see that the inventive finds in North America separate themselves out into 3 distinct clusters. So the first population that was found in North America is this population down in here. And then the population on the west coast is very, very distinct. And surprisingly, a year or 2 later, we found population or specimens from the northeastern part of the US, which was distinct from our mid-Atlantic population. And we were not expecting that at all. But this gives us a lot of confidence that we've had at least 3 separate introductions of Japonicus into North America. And then the Swiss population, again, is very distinctive. It clusters quite away from the other populations. And again, the Swiss population matches pretty well the Japan cluster. Our populations from North America, however, don't really cluster. Korea, all the Korea sites that we have are around here. Populations from China are around here. And then our US populations are not really close enough to either of the Asian clusters to allow us to identify to a high degree where they may have come from. So I've talked about all that. So in some of the implications of having an inventive population in North America, right away it gives us the opportunity to start doing field studies on what is the actual impact of these wasps on non-target species in North America. And this is something that researchers across the US are trying to work on now. We have to have large enough populations of wasps in the field, and this isn't yet the case in some areas, but we do have the opportunity to start to look at this now. And I mentioned the interest in moving the Sulcus japonicus from states where it does occur to places where it doesn't yet occur. In North America, our regulatory agency, APHIS, regulates interstate movement of exotic organisms, but it doesn't regulate intrastate movement. So once an inventive natural enemy is present within a state, the state regulatory agencies in that state can decide to allow it to be moved around so that it occurs everywhere in the state. And this is what most of the areas are starting to do. Well, let me backtrack a little bit. I'll get back to that in a moment. I wanted to say a word or two about how will these inventive populations of exotic species impact the native non-targets. Again, we have a range of successful parasitism with any species of inventive that's not perfectly adapted to a non-target host. It can be fully successful, leading to an F1 generation. Such as here, we have an F1 wasp ready to emerge. But at the other end, we can have unsuccessful parasitism. It may not recognize the host, it may not lay eggs in the host, or if it does lay eggs in the host, those eggs may not develop, or they may develop partially, but they may nevertheless kill the host. And this is what we have seen in our host range tests in the laboratory. And with the development of molecular markers, we also see this happening in the field. And some very nice work done by people like Tara Garapi in her lab in Canada has developed markers to distinguish parasitism by the various native Sulcus species. And that food web shows the complexity of what happens in a group of native stink bug species and mixed group of or complex of parasites that attack them. Can be very, very complicated. There was some thought that because Sulcus japonicus was not a successful host for some stink bug parasites that would attack it, like Telenomus pedicea, that brown marmorated stink bug represents an evolutionary dead end, but also a sink for eggs. Telenomus is a species, Telenomus pedicea is a species that seems to readily attack it in the field, but almost never develops successfully, so it's wasting a lot of its eggs. And this could have impacts on the population of Telenomus and the biocontrol of some of the other hosts that Telenomus parasitizes in nature. And with the ability of us now to detect what's actually going on with these molecular markers, we're able to try to decipher all these complex interactions in the field. Now we're starting to be at the point where we're gathering a little bit of information as to what actually happens in the field. In Washington, a nice study was done using sentinel egg masses of BMSB and 3 species of native non-target pentatomids. And these were all species that had been tested in the laboratory in the no choice and choice regime, showing successful levels of attack at various degrees. But under field conditions, when the egg masses were paired on the same host plant, the authors found a much lower rate of attack and development by Sulcus japonicus in the non-target species. And so this is an example of behavioral and ecological factors that further restrict the host specificity beyond what we see in the lab results. This is encouraging from a point of being able to argue that these parasites are exhibiting more specificity than would appear under the lab conditions. We've also been looking at the chemical ecology of their host binding and host acceptance. And a student at the University of Delaware looked at the host recognition by adult Sulcus japonicus wasps on leaves that contained the pheromones of either BMSB or an important North American predatory species, Podesis maculaventris. And this is a species of particular interest in the U.S. because it's regarded as an important beneficial species that feeds on other pest insects. It's a common predatory pentatoma that is found in agricultural settings. And it happens to be a species that Sulcus japonicus attacks and develops in quite well in the lab assays. But in the behavioral assay, the student Sean Boyle was able to show that Sulcus japonicus females do not, they recognize the host pheromone, but they respond more strongly to pheromones of BMSB than to pheromones of the Podesis. They respond by increasing their searching ability, which increases the likelihood that they'll actually find an egg mass. So again, this contributes to an additional degree of specificity under conditions that are more likely to occur in the field. And this work was extended by another graduate student, Robert Malik. He was able to show that the female Sulcus japonicus recognized not only the pheromones of female BMSB, but also male and nymphal BMSB. And in all of these cases, it responded more strongly to the pheromone of BMSB than to the pheromones of Podesis. In fact, it didn't even recognize nymphs of Podesis. It didn't change its search behavior at all. And Sean's work also showed that when he scaled up his work and put egg masses on plants in cages and reared Sulcus japonicus out of 2 different hosts. In this case, he had test females that had been reared from BMSB parents and test females that had been reared from Podesis maculaventris females. And if you remember, the females that come from Podesis maculaventris are smaller and perhaps less fit than the females that are produced from BMSB. In both of these cases, the females attacked BMSB in these cages much more successfully than finding and attacking egg masses of Podesis maculaventris. So just another indication that there's some behavioral specificity occurring. And Robert also was able to show that the active components of the pheromone that was deposited on the leaf surfaces seems to be due to 2 chemicals, a mixture of this E2-decanol and tridecane. And these are compounds that occur in a wide range of pentatoma actually, but they occur in differing percentages. And there are some minor peaks, very low amounts that could possibly be important in the blend. So back to this issue of adventive populations and aphis regulation. These are the states now that have begun active redistributions of Japonicus within their states, and some of the other states that aren't shown on here are preparing to release and spread Sulcus japonicus around. And APHIS has also recently told me that they're now willing to consider allowing Sulcus to be moved from a state where it does occur to a neighboring state where it doesn't occur. And up to this point, they had not been willing to make that decision. So I think the further distribution of Japonicus in North America is going to start to accelerate the dispersal. This gives an example of what we're doing in Delaware. This is our entire state. This is about 150, 200 miles from one end to the other, so we're one of the smallest states. And a postdoc, Joe Kaiser, is making releases at numerous locations, and he's making these releases as part of an experiment comparing the likelihood of establishment with a large release versus a smaller release. We don't have results back yet, but hoping to determine how many parasites are needed in order to guarantee the establishment and spread within an area. And this is being done in conjunction with some of the other states as well, so we have replicates in a number of different locations. And then this is work that derives from some of the work that Gonzalo and his team started last year in Delaware when they came over. And in southern Delaware, replicates were set up with this array of potted plants with sentinel egg masses, and then a central point release was made of Sulcus japonicus. And I'm not going to comment on the results of your releases. I don't know if you've talked about that at all, but I'll just say that at the end of the season we brought some of these pots back to our lab in northern Delaware, and we started an experiment to compare the success of attack on plants that have had stink bug on them with plants that did not have stink bug on. So on some of the plants we had cages where we caged brown marmorated stink bug for several days, let them walk around on the leaves, and then we took the cage off and we took the stink bugs off, and we put our sentinel egg masses out. So some of the plants had sentinel egg masses and no stink bug, and some of them had sentinel egg masses with stink bug. And the conditions in this, we were able to run one trial at the end of the season. Conditions were not ideal that day, but we did get recovery. So this shows a grid. Each circle is a plant that had a stink bug egg mass. The wind was blowing in this direction, and we got recoveries and parasitism of egg masses on all the plants in red. And we did have a higher response, a higher parasitism of the stink bugs that had, or the egg masses that had stink bugs walk on those particular plants. And we hope to look into this in much more detail this coming season. I think we were a little bit unlucky on these dispersal experiments this year because just about every day we had the releases, the weather conditions were not ideal. Strong winds and rather rainy and cool. So we need more replicates to see how they behave under more typical or a wider range of weather conditions. But it's clear that these parasites can move and they can find egg masses. We don't know yet how far they move, but from other experiments where stink bug, we've been monitoring stink bug in conjunction with various IPM schemes and putting out parasites. We do know that they will go from release sites outside a crop and they will move into the crop. And we've had recoveries inside crops, even crops that have been treated with insecticides. We have seen some parasitism, so we know the parasites are definitely capable of moving around. And I imagine you all know pretty much about this, the plans for the preemptive biocontrol of Sulcus japonicus in New Zealand, if and when brown marmorated stink bug populations are found in the field. This is a first in the world, as far as I'm aware, a great example of preemptive biological control. In Australia, they're also very concerned about brown marmorated stink bug. They haven't been as willing to consider preemptive release or even release of an exotic like Sulcus japonicus, and for the moment they're pinning their hopes on this other species of Sulcus. It's a curii that already occurs in Australia. But Sulcus mitsucurii is, although it will attack brown marmorated stink bug, we don't find it as a dominant member of brown marmorated stink bug communities in Asia, so it's too early, I think, to speculate on how it will do in Australia. And then finally, you're also aware of this idea that Kieran is going to be working on. the idea of extending sterile insect techniques to the parasites so that a sterilized natural enemy could be released, still capable of finding and killing the host, but without the capacity to develop itself, and thus not being able to establish in the environment, and no worry for extensive non-target impact. So this is a very interesting idea that hopefully we'll see how that will develop in coming years. We have a permit for field release of Sulcus Japonicus, the Beijing population in Canada. It's still in review, and planning to submit a petition soon for release of the Beijing population in the US. So in summary, we're just at the point of being able to start to gather important information about the impact of Sulcus in the field, and we're starting to, some results are starting to trickle in, but a lot more work needs to be done. And back to the idea of BMSB as a serious pest, I'll just very briefly give an update on some of the other management work that's being done in North America. When BMSB first became a big problem in North America, this is the kind of situation that growers had to respond to. This particular grower grows a number of different crops, and each color is a spray day for a different combination of crops. But you can see he's spraying every day for brown marmorated stink bug for some of his crops. So this would have been, well, that's June 2011. And the evolution of management has gradually moved away from frequent applications as emergency exemptions were obtained to work with a broader range of compounds, and lab tests were done to show, lab and field tests were done to show which pesticides were working and which weren't working, and additional tools were brought in. You can see that the use of pesticides gradually goes down, and the introduction of other techniques like pheromone traps for monitoring and other types of targeted spraying have been investigated in greater detail. There's just another example of changes in insecticide application, specifically in apples in Pennsylvania, showing the combination of OPs and carbamates and neonicotinoids and so on from the early days of brown marmorated stink bug to where we are now in 2019-2020. So you can see that the number of applications that growers are using is down, but it's still much higher than they would like to be putting on. And it has affected their IPM programs for other pests by causing secondary pest outbreaks and so on, where they weren't having to spray for other pests. Now they're having to spray for additional pests as well. So there's still significant additional costs for growers. So there's been a lot of work done also on traps for monitoring and for developing thresholds of management for BMSB, and the most effective traps are these pyramid traps, the black traps, the baited with the aggregation pheromone, and the bugs come to the black and then they crawl up into the trap. However, those traps are expensive, they're kind of unwieldy. Growers don't want to rely on them because they're just too difficult to manage. So a lot of work has been done to test simpler trapping regimes. And recently, multi-state trial was completed comparing the pyramid trap with this type of simple, clear, sticky trap panel that's baited with pheromones. So this clip here holds the sticky trap on, but there's also a pheromone on there. And it turns out that the black traps are more efficient in terms of giving better numbers, but the correlation of the numbers going to the sticky panels is pretty good. So these are methods that can be adapted for use by growers. And development of thresholds. And currently it's thought that a cumulative threshold of 4 adults on these sticky panels looks like a good target to shoot for, and they're testing this in commercial crops to see if it can be validated for use. And this is in apples. But similar work being done in other crops, such as peaches. There's been a lot of work done on the potential for border sprays. And as many of you know, a lot of insects have a strong edge effect when they're insects that move from habitat to habitat will often stop at the edge of a new crop, and this is definitely the case with brown marmorated stink bug. There is a strong edge effect. So there are a lot of recommendations are to spray only the borders of crops where they border habitats that the stink bug could be moving in from, such as neighboring wooded areas. Another technique, various crop crops have been experimented with, sunflowers for example. One of the most interesting techniques is the attract and kill method, where certain trees are picked and traps are put with pheromones to attract, and then those individual trees are sprayed. So this is a block of orchard trees, for example, and the red shows particular trees where the attractant pheromones are hung, and those trees are sprayed, and the trees next to them are sprayed. But the entire block doesn't need to be sprayed, and this saves a tremendous amount of time and effort and expense for the grower. And then finally, another interesting technique are the use of what are being called ghost traps. And this is netting, which has been treated with insecticides. And this is commercially available by some producers now, like Vestergaard. And these can be placed, hung around the perimeter of a crop, or placed on, well, this is just for test, I guess, but placed on panels like this. And these have been found to be very effective at killing the stink bugs, which when they come into an area, they tend to land on this screening first, and they'll pick up a lethal dose of pesticide and drop to the ground. So in summary, BMSB does represent still a serious challenge to farmers and homeowners, but a lot of research is being done in IPM aimed at finding ways of reducing the amount of pesticides that need to be used. And we're moving ahead on biological control, and hopefully in the near future we'll be able to get a better idea what the role of biocontrol can be in managing BMSB in stink bug in North America. And at this point, I'll let Elijah talk about the taxonomy of Sulcus. Then we'll come back and I'll just give a very brief overview of the other two tests.